Hello, welcome to World of Microbes. In our last video, we watched the cellular machinery pull off the ultimate copy-paste job, DNA replication. We saw how a cell duplicates its entire genome so that when it divides, both new cells get a perfect, identical blueprint. It's an incredible feat of biological engineering. But think about it, your cells don't need to build a whole new cell every single second. Most of the time, they just need to use those instructions to actually do something like create a digestive enzyme, build muscle fiber, or manufacture a hormone. The master blueprint never leaves the safety of the nucleus. So, how do those instructions get out to the cellular factories? Enter DNA transcription. Transcription is the precise process of copying a single, specific gene into a portable, temporary message called RNA. Today, we're going to break down exactly how your cells unzip just a tiny fraction of that DNA, rewrite the code from T to U, and create the ultimate biological memo. Let's get into it. Before we look at the mechanics, let's define exactly what we are dealing with. By definition, DNA transcription is the biological process where a specific segment of DNA is copied into a complementary strand of RNA. It is the critical first step of gene expression, essentially. It is how your cells read their instructions. Think of your DNA as a massive, priceless encyclopedia locked safely inside the cellular vault, the nucleus. The cell cannot risk damaging or losing this master copy by sending it out to the busy factory floor. Instead, it creates a temporary, lightweight photocopy of just one single page. That photocopy is messenger RNA, or mRNA. Here is the high-level overview of how this happens. A specialized enzyme called RNA polymerase locates the exact gene the cell needs. It unzips that specific section of the DNA double helix and reads one of the strands as a template. It then matches free-floating RNA nucleotides to that template, weaving them into a single-stranded message. The chemical language is almost identical, with one major twist. While DNA uses the bases A, T, C, and G, the newly formed RNA replaces every thymine with uracil, pairing you with adenine. Once the gene is fully copied, the RNA strand detaches, the DNA zips back up perfectly unharmed, and that fresh RNA molecule is ready to carry the instructions out into the cell. While DNA replication and transcription look similar on the surface, they serve completely different purposes, use different enzymes, and produce entirely different results. Let's stack them head to head. First, the goal and scale. DNA replication is an all-or-nothing event. It copies the entire genome, from start to finish, exactly once. The goal is to prepare a cell for division, ensuring the new cell gets a complete set of DNA. Transcription, on the other hand, is highly selective. It only copies a single gene, a tiny fraction of the DNA. It happens continuously throughout the cell's life whenever the cell needs to make a specific protein. Second, the machinery. Replication relies on DNA polymerase to build the new strands, and it requires a primer to get started. Transcription uses RNA polymerase, which can start building completely from scratch without a primer. Finally, the output. Replication yields a permanent, double-stranded DNA molecule utilizing the base thymine. Transcription yields a temporary, single-stranded RNA molecule that replaces thymine with uracil. Now that we know the differences, let's look at exactly how transcription works. The entire process takes place in three tightly controlled stages: initiation, elongation, and termination. Let's start with stage 1, initiation. Now that we know the differences, let's look at exactly how transcription works. The entire process takes place in three tightly controlled stages: initiation, elongation, and termination. Let's start with stage 1, initiation. This brings us to stage 2, elongation. RNA polymerase reads just one side of the unzipped DNA, known as the template strand. As it moves along, it builds a complementary strand of RNA by matching free-floating nucleotides to the DNA template. Remember our base pairing rule change from earlier. Adenine on the DNA now pairs with uracil on the growing RNA strand. The enzyme keeps moving down the line, lengthening the RNA chain until it hits the final stage, termination. At the end of the gene sits a specific sequence of DNA called the terminator. This acts as a molecular stop sign. When the enzyme crosses the terminator, it receives a signal to release. The RNA polymerase detaches, the freshly minted RNA transcript floats free, and the DNA strands snap back together perfectly unharmed. Transcription is complete. While the fundamental goal of transcription is the same across all life, the execution is vastly different depending on whether you are a simple bacterium or a complex multicellular organism. Let's break down the major differences between prokaryotic and eukaryotic transcription. In prokaryotes, like bacteria, there is no nucleus. Everything happens in the fluid cytoplasm. Because there are no physical barriers, translation can begin while transcription is still happening. It is a rapid, simultaneous, all-in-one process. Eukaryotes, like humans, have a strict boundary. Transcription happens exclusively inside the nucleus, safely locked away. The resulting RNA must then be heavily processed and exported out to the cytoplasm before translation can even begin. The two processes are completely separated by time and space. Prokaryotes keep it simple with just one single type of RNA polymerase to handle all transcription. Eukaryotes, however, require specialized labor, utilizing 3 distinct RNA polymerases labeled 1, 2, and 3 to build different types of RNA. Prokaryotic RNA is functional the exact moment it is made. It needs no editing. Eukaryotic RNA is born as pre-mRNA and requires major modifications. It must get a protective 5-foot cap, a protective poly-A tail, and undergo splicing to cut out non-coding sequences before it can ever be used. So let's talk about post-transcriptional modification in details for better understanding of transcription in eukaryotes. When eukaryotic transcription finishes, the resulting RNA molecule isn't quite ready for the real world. At this stage, it is called pre-mRNA. It is incredibly fragile and full of genetic junk. Before it can leave the nucleus safely, it must pass through an intensive editing process called post-transcriptional modification. Step 1 is 5' capping. A specialized, modified guanine nucleotide is attached like a hard hat to the front or 5' end of the RNA strand. This cap serves 2 critical roles. It protects the delicate RNA molecule from being chopped up by cellular enzymes, and it acts as a recognition handle so ribosomes know exactly where to attach later on. Step 2 happens at the tail end, polydenylation. A long chain of hundreds of adenine bases, called a poly-A tail, is tacked onto the 3-foot end. Think of this tail as a protective buffer or a ticking fuse. The longer it is, the longer the RNA survives in the cytoplasm to make proteins before it naturally degrades. Step 3 is the most dramatic edit, RNA splicing. Eukaryotic genes contain non-coding junk segments called introns, interspersed between coding regions called exons. A massive molecular machine called the spliceosome cuts out the useless introns and pastes the remaining exons tightly together. It literally splices the functional code back into a continuous story. Once the pre-mRNA is capped, tailed, and spliced, it officially becomes mature mRNA. It is now stable, streamlined, and cleared for export out of the nucleus and into the cytoplasm, where the factory floor is waiting to translate it into a protein. The editing room is closed. And there you have it. The cellular master blueprint has been successfully accessed, safely copied, and flawlessly edited. What started as an unbending strand of DNA locked inside the nucleus is now a mobile, highly stable message. Mature mRNA, out in the cytoplasm and ready for action. But a message is only useful if someone can actually read it. Right now, this mRNA is carrying a code written in nucleotides, but the cell speaks the language of amino acids and proteins. To bridge that gap, the cell has to deploy its ultimate manufacturing factories. How does that code get cracked, and how do those letters transform into the physical proteins that build life? That is exactly where the story goes next. Thank you so much for watching, keep questioning, and we will see you in the next video for DNA translation.